Well, first of all, I want to welcome everyone here and to express my thanks and gratefulness to Dr. Su, who gives me the chance to explain to you the basic mechanisms which underlie my concept and the treatment. I have separated two parts today in my lecture. The first part is about the infusion protocols, and please understand these protocols can be combined as a complementary treatment with anything you're doing in your own special field of medicine. And we can discuss in which way you would optimize it. I want to outline the pathomechanisms of a lot of different diseases which are associated with chronic inflammation. And I want to explain to you why this is a general feature in aging patients. I start. I think our aim is to, in the first place, to prevent diseases and to promote health mechanisms in our body network. So why is it that we age? What are the main causing factors which drive us, our body, our regulatory networks to age? I think it's not that difficult and complex if we look at it. A very important feature is our immune system. So it has been demonstrated for quite different types of diseases that the balance of a homeostatic immune system skips and causes chronic inflammatory responses in various organs of the body. And this ongoing chronic inflammation then is a very important contributor to disease manifestation. This has been shown for neurodegenerative diseases, for diabetes, liver diseases, muscle and vessel disease, coronary heart syndrome, stroke. In all these instances, I will show you it is the immune system which is a driving force. So my concept is aging basically relates to the ability of our stem cells to repair and to restore the body integrity and natural body functions. If we block our stem cells in various stem cell niches, we have specific stem cells in the skin, in the gut, in the liver, in the muscle, in the brain. And if you block those, then you will have a lack of regeneration, and this causes aging. But in order to have an optimal stem cell function, you also need to balance your immune system, your endocrine system, and metabolism. And because all these four components play together in our body, it's not just to look at the stem cells. You may inject stem cells of whatever source. It may be mesenchymal, hematopoietic, pluripotent stem cells. If you inject your stem cells into an acid environment, they won't work. So you have to regulate the environment before you treat with any cellular product. So for me, the underlying pathomechanism is based on chronic inflammation, tissue acidification, dropping hormone levels, impaired immune activation. And only if you keep in mind all these four components, you will be successful in rejuvenation treatment. It has been published a couple of years ago that inflammation is closely related to diabetes and a lot of different metabolic disorders. Just to read here, obesity is associated with an array of additional health problems, including increased risk of insulin resistance, type 2 diabetes, fatty liver disease, atherosclerosis, and degenerative disorders. Why is this? Well, if we have an oversupply of nutrition, at one point, more and more free fatty acids are released from the adipose tissue, and this causes adipocyte hypertrophy, macrophage recruitment and activation, and the overstimulation of our visceral fat causes an immune activation which will result in a systemic immune insulin resistance and inflammatory response of these infiltrating immune cells. Here you see the yellow cells, these are macrophages which activate the immune system, and then this will also disturb the metabolism in the liver and in the muscle. I will come to that point in more detail in a second. So basically at this point what I'm saying is a disturbed metabolism mainly due to overnutrition in fats, sugar, carbohydrates can cause an activation of immune cells which produce inflammatory cytokines, a certain chemokine receptor expression which will recruit T cells and other lymphocytes to the site of inflammation. And this has been shown, for instance, to cause atherosclerotic plaques, but also it is closely related in diseases like coronary heart disease, stroke, diabetes, neurodegenerative diseases, psychological disorders, and last but not least, can cause cancer. I can tell you anything, but I have to prove it that this is a hypothesis which has also been registered by other colleagues in the field. And that nutrition is closely linked to disease formation is not something I have invented. There is abundant knowledge and publications which indicates that certain intermediates which are produced from glucose and lipids can cause these diseases. And one of the major threats of our nutrition is the formation of advanced glycation end products. What are these? Advanced glycation end products are a heterogeneous group of bioactive molecules which are formed in a non-enzymatic glycation of proteins, lipids, and nucleic acids. And these AGs have been implicated in the pathogenesis of diabetes, atherosclerosis, renal disease, and other, for instance, Parkinson and neurodegenerative diseases. How can you visualize that? If you have a protein string, there are amino residues which can cross-link with either sugars, sugar moieties, or lipid moieties, and these can cross-link the protein folding, so the functional features of these three-dimensional proteins are disturbed, and they will stimulate wrong metabolic and enzymatic activities. In the other place, some of these carbohydrates and lipids, when they cross-link to the proteins, they form so-called glycotoxins, and these have been reported to directly induce complications in the kidney and atherosclerosis. So there is a certain paradigm shift, as has been published in Nature Reviews Endocrinology, from concept of mere overnutrition, adipose tissue obesity, insulin resistance, and then all the metabolic problems. Today we think that it is the food we take and the content of these advanced glycated end products which induce an impaired immunity with consequent simultaneous inflammation, and that inflammation causes direct tissue damage in the vessel wall, cartilage, connective tissue, and so on. That these products have a direct clinical relevance, and a very important clinical relevance, has been published in a couple of clinical trials, and this is one of Johns Hopkins University in Baltimore, which I just wanted to disclose to you as an example. They prospectively tested 1,002 women in this study, and they measured one of the most common age products, the carboxymethyl-lysine, which is a carbohydrate moiety which binds to a protein. And we have, this was developed in Germany, an ELISA method to determine the level of CML in the blood of the patients. So the outcome of these women monitored for 5 years is shown here. Here you see the survival curves for all-cause mortality among women above 65 years, where they measured the amount of CML in the serum of these patients. And women in the highest quartile, seen here, depicted here, had a significant lower survival than women with less CML in the blood. And this was quite significant finding. So you see about 30% of women don't live for about 5 years. They die within 5 years if they have a certain threshold of CMLs in the blood. If you look for the survival of cardiovascular disease mortality in the same cohort of women, it had been even more significant that these women with high CML levels had a significant lower life expectancy and more cardiovascular disease mortality. So I think this very convincingly demonstrates the impact of nutrition, the consequences of inflammation, and for disease development and mortality. So what really happens is that these low-density lipid particles can enter the vessel wall. You see, this depicts an arteria. Here's the lumen, the cells floating. We take up low-density lipids, and these H components, they promote the oxidization of these low-density lipids, and then these oxidized LDL particles are scavenged by macrophages as an innate immune cell, and the internalization of these particles activates certain signaling pathways which causes chronic inflammation, and then this is the first step for atherosclerotic plaque formation. It has been demonstrated in animal experiments of diabetic NOD mice that if you compare a low and a high HCHO food given to these animals, you have quite an interesting finding in terms of kidney and vessel formation of these products. You see here, this is after 2 months, an animal fed with high HCHO versus animals with low CHO H, and you see the comparison at 5 months. It's really interesting how the formation in the glomeruli The deposits here you can see in comparison to the other group. And this explains also the diabetic complications we find in kidneys in Wilson disease with the apparent glomerulonephritis. And if you look at the vessel, here the experiment was, they looked at the femoral artery of these rats and they made an injury in the intima, and then they compared the outcome after 4 weeks. So 4 weeks after the initial injury, they compared animals with low and high age food. And here you see low and here high nutrition age and the obvious stenosis in animals, same genetic strain, but a very different outcome, merely depending on the level of these end products, glycation end products. And last not least, it has been demonstrated very convincingly in this paper published in Cell that inflammation is associated with neurodegenerative disease, including Alzheimer, Parkinson, amyotrophic lateral sclerosis, and multiple sclerosis. And it's basically the same mechanism underlying the formation of mutated proteins, as depicted here for the example of Alzheimer. So these glycotoxins, they activate or they downregulate a certain, this is a surviving protein in the brain cells, which affects the insulin receptors substance. This is a sensitizer for insulin, and if you downregulate this protein, then the cell is not able to take up the insulin anymore, the brain cell. You have a brain-specific insulin resistance, which is always associated with then type 2 diabetes. So Alzheimer's disease and type 2 diabetes are closely connected. On the other side, the impairment of this protein will increase the formation of this mutated protein, the tau protein, and will phosphorylate it and induce directly neuronal toxicity, which causes cognitive impairment. And of course, inflammation supersedes the whole mechanism. So there is a metabolic link to the formation of these proteins and disease induction. And this depicts that our microglia cells, which are the correlate immune cells in the brain, they have a receptor for H. So these products are bound to this receptor, and this causes the transcription of inflammatory cytokines like TNF, interleukin-1 beta, which also affect the metabolism in the astrocytes, which are in the vicinity of these neurons. And both cells, astrocytes and microglia, can be triggered to produce radical oxygens, nitro oxide, inflammatory molecules, which then impair and endanger and injure the motor neurons and other neurons, which causes the Alzheimer and other neurodegeneration. In cancer, it is very clear and has been demonstrated also that the acidic extracellular pH is a major feature of the tumor tissue and considered to be due to lactate formation from anaerobic glycolysis. And this has a strong impact on tumor aggressiveness and metastasis, which I'm going to show to you. This is another paper which clearly states that tumor acidity trends to correlate with cancer aggressiveness and can, if you regulate it in a way that you lower the extracellular acidity in the vicinity of the tumor cell, this protects from from cancer formation and aggressiveness. One of the molecules, and this is also part of our protocol, which I will explain to you in a second, is called dichloroacetate. And this molecule has a very potent ability to normalize this lactate formation and tumor-related acidity. How does this work? So, coming back to the biochemistry of glucose metabolism, you know that under aerobic conditions, glucose is converted by an enzyme called pyruvate dehydrogenase, PDH, converts glucose into pyruvate, and pyruvate is the most important molecule for the mitochondria to produce ATP as the energy forming unit which all mechanisms in our body need. So under normal conditions, one glucose molecule is converted to 36 ATP molecules. If the glucose metabolism is disturbed because this enzyme is blocked, then we don't form pyruvate, we form lactate. And then from a lactate formation, the cell produces only 2 ATPs. So how can we envision that this happens in a cancer or a hypoxic situation? Well, if a cancer cell grows, the arterial supply with oxygen in the center of the tumor tissue creates a hypoxic state, because the cancer is growing faster than the arterial supply can go on. And in that situation, and this is also true for any other hypoxic situation, in hypoxic environment, a factor is formed which is called HIF. And this stands for hypoxia-inducible factor. And this factor can impact on glucose transporter. It will bring up more glucose in the tumor cell, and it activates an enzyme, pyruvate dehydrogenase kinase, which blocks the PDH. So these factor actually contributes to inhibition of the normal metabolic pathway, and at this time you don't build pyruvate, but the tumor cell just converts to lactate. And this has an impact on the mitochondrial ATP, as I explained to you, and it also has a very strong impact in the sense of a tumor escape mechanism, because if you have a lot of lactate in the cell, at one point lactate is released to the vicinity of the cell outside in the extracellular space, so this makes it acid. In turn, calcium and potassium are inserted into the cell, and they don't allow the mitochondria